Well, good morning or good afternoon, everyone, wherever you are in the world, and welcome to the 2019 Year in Review, where we'll be highlighting key publications utilizing the RNAscope technology. My name is Courtney Anderson, and I manage the applications group here at ACD. So it's been a really stellar year for publications with RNAscope. There were over almost 800 new peer-reviewed publications in 2019, giving the total number of publications using RNAscope up to 2,646 papers. And there's some really stellar work that was presented this year. In addition, these papers have also been in very high-impact journals. Over 25% of all of the RNAscope papers, or papers using RNAscope, have been published in one of the big three publishing house journals, so one form of Nature, Cell, or Science. So very high-impact papers. And for research areas, we have two really major research areas that are represented in the publications, neuroscience and cancer by far, but also infectious. So with those top research areas, we'll be highlighting a number of those papers in this presentation today. Just first, to give a high-level overview of the RNAscope technology. This is an ideal spatial analysis solution to interrogate complex tissues. It's a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single-cell level. And it consists of three parts. A unique target probe that we depict as these double Zs that ACD designs against your sequence of interest, a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots, which lends itself for quantification. And this can be done on either chromogenic or fluorescence assays. And it really is ideal for the spatial mapping of almost all RNA species, including messenger RNA, long non-coding RNAs, splice variants, highly homologous sequences, and even point mutations. And the assay can be performed on a wide variety of sample types, including FFPE tissues, fresh or fixed frozen tissues, and cultured cells. Now, the assay, there are two unique assays that make up the RNAscope technology. The main assay that is the RNAscope assay. It's the bread and butter assay. This covers most RNA species, really messenger RNAs and long non-coding RNAs that are greater than 300 nucleotides in length. And the probes are approximately 20 of these double Z pairs, targeting around 1,000 nucleotides. We also have another assay called BaseScope. This is a more specialized assay that really helps interrogate really unique RNA species that have not typically been able to be interrogated in the tissue context in the past. With this assay, we now can get down to as few as 1 to 3 double Z pairs, so we can target as short as a 50 nucleotide sequence. And this really now allows researchers to interrogate exon junctions, splice variants, exon skipping, any short or highly homologous sequences, such as the CDR3 sequence from T-cell receptors or MAGE family members, and even down to point mutations, such as a KRAS point mutation. So we really, between the two assays, are really able to get spatial mapping of almost all gene expression at the RNA level. Now, this is just a broad overview of the RNAscope and BaseScope product portfolio. We offer the assays both in chromogenic as a brown or red assay. You can do them as a duplex chromogenic with red and green. With BaseScope, that is a chromogenic assay, also offered as a single or duplex assay. And then with our multiplex fluorescent line, we do offer this in a couple of different flavors. We have one that is really specific for fresh frozen tissues. It can interrogate up to 3 targets. Our multiplex V2 assay is really has been optimized to detect gene expression in FFPE tissues and get around some of the autofluorescence issues with those tissues. And that can detect up to 4 targets. And then our most recent assay is the high-plex assay, which can detect up to 12 targets now in the tissue context. In addition to the assays, we also have a number of probes. We have over 22,000 probes that are already in our catalog. But if you cannot find your gene of interest or against your species of interest, any probe can be designed against any target from any species, provided the sequence is known, in as little as 2 weeks. Now, one assay that I did really want to highlight, we're very excited about this, this was launched in 2019, is our new high-plex assay. It's a very powerful assay that now allows spatial mapping of up to 12 targets in the tissue context. It is an iterative fluorescent detection in groups of 4 targets at a time. So it works essentially like the standard RNAscope assay, where you would hybridize with all 12 of your probes. You do signal amplification, but the labeled fluorophore happens in rounds of 4 targets. So you would do your first 4 fluorophore hybridization. You would take a picture or image it under the microscope. You then cleave off just the fluorophore. You're not cleaving off any of the probes or anything else that's part of the sequence. It's just the fluorophores that get cleaved off, which is a very important point. By cleaving just the fluorophore, there's minimal to no effect on the RNA or tissue morphology. After you cleave off those first 4, you then hybridize it with the next 4 fluorophore labels, so for channels 5 through 8. You take another picture, and then if you're continuing on with up to 12 targets, you would do that last round of 4 targets. And then there is an image registration software that comes along with the assay that will stitch all 3 of those images together. And this image registration algorithm is really able to precisely enable true 12-plex data visualization at the single cell level. And this assay is compatible with both fresh frozen and fixed frozen sample types. And really it's ideal not only if you have a large number of targets that you want to look at, but also if you have precious samples. So you can run more targets but on fewer sections. Or if you're working in a tissue such as the brain, where as you section through the tissue, you very quickly change structures. This is just one of the examples of some of the high-plex assay data. This is 12 targets that we were looking at in the mouse striatum to characterize the DRD1 and DRD2 subpopulations. And you can see nicely as the image moves, you can start to see cells become positive for the various different genes that each color is representing here. And this is just a still shot of that image. So you can see very nicely the detection of all of these 12 targets at the single cell level. So with that, I'd like to focus now all on the highlights of 2019 for our publications. And so we've divided this into several sections based on those key research areas. And the first we wanted to start with, because we felt high-plex is a very strong assay to help with single-cell RNA sequencing analysis for spatial mapping and confirmation. And so we wanted to highlight several of the papers that were published this year for this reason. This is the first paper. Now this is coming from Sarah Teichmann's lab and the Human Cell Atlas. This is a seminal paper that was published in Nature Medicine that described a cell atlas of the healthy human lung as well as the asthmatic lung. And with regards to asthma, they used more than 25,000 cells from both healthy and asthmatic patients to reveal mucus cell hyperplasia in the asthmatic lung. And the multiplexing capabilities of the RNAscope assay allowed for confirmation of a strong increase in mucus-ciliated cells in the asthma lung by detecting co-expression of MUC5AC, which is a secretory marker, as well as FOXJ1, which is a ciliated cell marker. Now, this is another very nice paper that came out of the Broad Institute at MIT. They really wanted to understand the cell type specificities and pathways of action in ulcerative colitis. So these researchers created an atlas of cells from the colonic mucosa of both healthy and ulcerative colitis patients. And they analyzed over 350,000 cells from these patients. And they revealed through their single cell analysis over 50 epithelial, stromal, and immune cell subsets. And the researchers combined the RNAscope fluorescent assay with IHC to validate and spatially map all of the major findings that came out of the all the major cell types that were identified by these single cell analyses. So this is just to show one example of that. This is looking at the characterization of BEST4 positive epithelial cells, as well as respondin-3 positive fibroblasts in the healthy colon. So they identified BEST4 as being a marker of epithelial cells, as shown here by colocalization with the EpCAM epithelial cell marker. And then respondin-3 was a marker of the fibroblast, as shown by colocalization with fermentin, which is a fibroblast marker. And then on the right side, they showed an increase in inflammation-associated fibroblasts in the inflamed ulcerative colitis tissue. So you can see on the right side of the screen that there's an increase in IL-13RA2 and fermentin double positive cells, indicating that there are these inflammatory-associated fibroblasts in the ulcerative colitis tissue. And this is a paper that just came out a few weeks ago, and it's a really powerful story showing how single-cell RNA-seq can now reveal subpopulations of hepatic stellate cells. It was typically previously thought that these were just a homogeneous population, but by using single-cell RNA sequencing, the authors revealed two subpopulations of hepatic stellate cells. They found that there are portal vein-associated HSCs as well as central vein-associated HSCs. And again, these researchers also combined the RNAscope-ish assay with immunofluorescence on the same section in order to reveal zonation of these subpopulations across the healthy liver. So you can see here on the left side of the image, the left side of the image is all immunofluorescence showing that NGFR is a marker of the portal vein-associated hepatic stellate cells. And then on the right, they used RNAscope to detect the ADAM-TSL2 marker, which they found to be a marker of the central vein associated hepatic stellate cells. And you can see very nice co-localization of the two genes in the two markers in the pericentral region. Now, furthermore, when they looked at a liver injury, they found that collagen 1A1 seems to be it's upregulated following liver injury, and that it appears that the central vein-associated hepatic stellate cells are the cell types that are predominantly producing collagen following liver injury. So they had identified this by the single-cell sequencing analysis on the left side of the screen, and then on the right, they again combined dual-ish with IF to show that collagen 1A1 is indeed colocalizing with the central vein marker, central vein associated hepatic stellate marker ADAM-TSL2, but it was not colocalizing with the portal vein marker NGFR. Now, this was a recent study that came out looking at the human retina. So the researchers here created a single-cell atlas of the human retina to reveal heterogeneity in macroglia cells. Now, age-related macular degeneration is one of the leading causes of blindness, yet its genetic complexity has really made it challenging to identify the cell types associated with disease. So these researchers performed single-cell RNA-seq on human retinas to really create the first single-cell transcriptomic atlas of the human retina. And that's where they identified this high heterogeneity with macroglia. And as you can see here on the bottom, they identified three distinct macroglial subsets by single-cell RNA sequencing. They list here the genes that were associated with each of those three subsets. And then they used RNAscope combined with immunofluorescence for a marker of glial and astrocyte cells to show the distinct macroglia subsets and to confirm that these genes are truly marking those three subsets. And these authors recently did a webinar with us, so please check the link below on the screen if you'd like to learn more about this publication. Now, finally, for this section, I wanted to highlight this publication, which created a very nice atlas of vagal sensory neurons. They're necessary for the control of organ functions, but the knowledge of the complexity of these neuron types really is lacking. So researchers from the Karolinska Institute present a comprehensive identification, classification, and validation of the neuron types in the jugular and the nodose-derived vagal ganglia. And they identified 6 types of jugular neurons as well as 18 types of nodose neurons. So here on the screen, you can see that there are distinct populations marking the nodose, which they show here with FOX2B as the nodose marker, and PRDM12 in green as the jugular marker. And then they show on the right a heat map with 330 differentially expressed genes between the nodose and the jugular clusters. And so they used RNAscope quite extensively in this publication, but here I'm just highlighting the nodose neurons. So they identified 18 different neuronal clusters as nodose neurons, and that's what's shown by the single-cell Tisney plot on the left. In the middle, for every column, that's each of those 18 clusters, and then every row is listing the genes that are expressed in those clusters. So they then went on and used RNAscope to validate at a single cell level the genes that are expressed in those clusters. So again, with the RNAscope data, every column is a different nodose ganglion cluster, and then every row is a gene. And so you can see very nice single cell detection of these genes for these cell types. Now I'd like to move on and highlight the publications that were really intriguing regarding neuroscience specifically. So this is a paper that came out just about a month or so ago looking at sleep and basically the transcript and proteinome related to sleep. So sleep is obviously very essential for mammalian brains, and deprivation of sleep can really result in significant cognitive impairment. The ability to sleep really is governed by both daily rhythm, which we know is the circadian clock, and also by the brain's need for sleep, which is referred to in the paper as sleep pressure. And that sleep pressure gradually increases the longer we stay awake and don't sleep. However, little is known about how sleep can restore brain function or how the need for sleep is really tallied. So these researchers provided a comprehensive overview of the daily changes in the synaptic transcriptome and proteome in the mouse forebrain. They evaluated messenger RNA and protein abundance over 24 hours in both normal and sleep-deprived mice. They found that the oscillations of 75% of the synaptic proteins were concomitant with their rhythmic mRNA transcripts. So this indicates a key role for local synaptic translation. They found that the transcriptome is really organized by the clocks, whereas the proteome is driven more by sleep. So this study really provides insight into the connectivity between sleep and circadian rhythm. To verify their transcriptional results and to distinguish between whether or not this abundance or the oscillation in the transcripts arises from either the transport of messenger RNAs or from the local control of their stability, the researchers used RNAscope. They showed that there was a spatio-temporal post-transcriptional regulation of mRNAs for genes that have been reported to have synaptic function. So shown here is one example, SLC17A7. They show that there's a diurnal abundance in the brain of this gene. They also quantified the synaptic signal cycling of these messenger RNAs. So a really intriguing study here. Now, it's a publication that identified a gene that's really required for the generation of pain-initiating neurons. The sensation of pain, itch, and temperature are really all essential for animal survival. However, the key molecular regulators that are necessary for initiating this pain-sensing development are really unknown. And so these authors identified the transcriptional regulator PRDM12 as being essential for pain perception. And they used the combination of ISH and IF to show that PRDM12 is expressed in the developing dorsal root ganglion. And they also show which cell types it is expressed in. So PRDM12 is expressed in all of the TRKA positive nociceptive neurons, but it's really expressed to a smaller extent in TRKC proprioceptive neurons and is not expressed at all in the TRKB positive mechanoreceptor neurons. So it shows that cell type specificity of this critical gene. There's another interesting paper also looking at pain, but this is focusing on chronic pain and the emotional component. Pain is really a multidimensional experience, and how much pain is bothersome to one person versus another, and it can also significantly impact the patient's quality of life. The Kappa opioid receptor, the Kappa opioid system, which functions through the Kappa opioid receptors, contributes to depressive and dysphoric states, but whether it contributes to the emotional component of chronic pain had remained tenuous. So these researchers show that the Kappa opioid receptors are sufficient to drive the emotional part of chronic pain that is argued to significantly impact a patient's quality of life. And the multiplex assay was used to examine Kappa opioid receptor expression in a mouse model of chronic pain. They did a peripheral nerve injury, PNI, and then they connect that expression to function. So you can see here on the left-hand side, they identified that the Kappa opioid receptor 1 is increased in the PNI mouse model and is primarily localized to TH-positive dopamine neurons and not expressed in the glutaminergic GAD-positive neurons. And then on the right, you can see here in the PNI mouse model that activity of the Kappa opioid receptor system is increased in the midbrain in these animals. There's an interesting paper as well looking at splice variants. The brain has really one of the highest amounts of splice variants compared to any other tissue, which really lends a lot of heterogeneity to the brain. And these researchers were interested in looking at a particular gene, the calcium 2.2 channel, and they were looking at CACNA1B, which encodes the pore-forming subunit of these channels. Now, there are many splice variants of this gene, and they are expressed in specific subpopulations of the brain, and they can influence channel function and sensitivity. But prior to the development of the BaseScope assay, there was really no way to see where these splice variants are expressed. to get the cell type specificity and to really interrogate and get that descriptive analysis of the splice variant. So these researchers used the BaseScope assay to interrogate one particular isoform, E37A, which whether inclusion or exclusion of this exon will lend itself to different splice variants. And you can see very nicely the detection of this splice variant in various regions of the mouse brain. Now, I want to focus a little bit more specifically within the neuroscience realm on neurodegenerative diseases. We had, again, a number of very nice publications utilizing the assay to interrogate neurodegenerative diseases, both in animal and human patients. So here we want to focus on this publication that was looking at Alzheimer's disease, which obviously we know is an age-related neurodegenerative brain disorder, but the molecular and cellular complexity really remains poorly understood. Now, both tissue analyses have indicated the downregulation of neuronal functions and upregulation of innate immune responses, but they likely have masked the complexity of the alterations across cells and within cell groups. So these researchers from MIT analyzed over 80,000 single nucleus transcriptomes from the prefrontal cortex of 48 individuals with either no pathology or with varying degrees of Alzheimer's disease pathology. This study really provides the first blueprint for interrogating the molecular underpinnings and cellular basis of Alzheimer's disease. And the RNAscope duplex assay confirmed the cell type specific downregulation of differentially expressed genes. They show here that there were significantly fewer excitatory neurons that were marked by SLC17A7 that also had detectable NTNG1 expression in brain sections from the AD patients compared to the no pathology patients. Now, this is a really interesting paper that came out looking at, in the mouse, a model of Alzheimer's disease and amyloid beta plaques. Now, we know tissues affected in many age-related diseases, such as Alzheimer's disease, can also exhibit accumulations of senescent cells. However, it's not known whether cellular senescence is involved in the pathogenesis of amyloid beta plaques. And it can be associated with cognitive impairment commonly seen in Alzheimer's disease. The researchers from the NIH found that oligodendrocyte progenitor cells in the amyloid beta plaques exhibit a senescence-like phenotype in the brains of patients with AD, and they also examined this in a transgenic mouse model of AD. And so using, again, the RNAscope assay combined with immunofluorescence, which is detecting the amyloid beta protein, they found that a marker of senescent cells, P16, is accumulating in the amyloid beta plaques of brains from the Alzheimer's disease mice. And so I'll just go back and start that video again so you can see the nice 3D digital reconstruction of the tissue samples that they used to stain with RNAscope in green for P16, and then the amyloid beta plaque is highlighted in red. They then went on to analyze the effect of 2 FDA-approved senolytic compounds to see if they could induce death of the senescent cells in the amyloid beta plaques. So again, by combining RNAscope with immunofluorescence, you can see after treatment, so the D plus Q image, you can see a reduction in the green signal, indicating that the P16 positive cells were being killed by the senolytic treatment. And so this is a potential path for preclinical and clinical studies, looking at senolytic therapies in neuropathology for AD and other related neurodegenerative disorders. Now, in this study here, this one was examining multiple sclerosis and looking at demyelination. As we know, it can lead to neurodegeneration, but the severity between patients is highly variable. And one factor could be because of oligodendrocytes, since the oligodendrocytes myelinate the axons. But until this study, the extent of human oligodendrocyte heterogeneity and its contribution to MS pathology had remained unknown. So these researchers analyzed single nuclei, took advantage of single nuclei RNA sequencing to assess postmortem human brains and revealed oligodendroglial heterogeneity in the MS patients. They performed the BaseScope duplex assay to complement their single nuclei RNA sequencing data and revealed that there was a correlation between the single nuclei RNA sequencing data as well as the ISH subcluster marker expression in the white matter of human control brain. So you can see here that there's very little overlap between subcluster markers. So they have KLK6 which is marking the oligo 5 cluster. CDH20 is marking the oligo 1 cluster, and there are no cells that are co-expressing both of those markers. Similarly, with the oligo 6 and oligo 2 markers, there's also no overlap where cells are co-expressing both of those markers. So this does indicate that these are distinct subpopulations of oligodendrocytes. Excuse me. So then if we look at the human MS brain, here they identified a unique population, this hybrid population that they called immune oligodendroglial cells. They are expressing both canonical oligodendrocyte genes as well as genes associated with macroglia, so hence they were called oligodendroglial cells. And again, they combined BaseScope-ish assay for CD74, which is a marker of these olig-1,2 positive oligodendrocytes. It's a marker of the microglial cells, and they combined it with the olig-1,2 antibody for IHC, and by showing co-expression of these two markers, they do indeed that there is this hybrid cell type of oligodendrocytes and microglia in the human MS brain. And then lastly, I want to focus on this section, again, another really interesting finding or discussion about a unique molecule considered paraspeckles. So paraspeckles are these subnuclear bodies that get assembled onto the long non-coding RNA NEAT1. And it seems that FUS is an essential component of paraspeckles. And it seems the dysfunction of these paraspeckle components has been identified as a pathological marker of many diseases, including diseases such as ALS. And so mutations in fused gene can cause ALS, but it's not known how the endogenous levels of mutant fuse would affect these NEAT1 paraspeckles. So these authors used RNAscope to look at the accumulation of the paraspeckles by using a probe for both NEAT1 and 2. And then they looked at these, this long encoding RNA both in healthy human spinal cord cells as well as in patients that have ALS and the fused mutation. And you can see here that there's certainly an increase in the detection of these paraspeckles, both in the neurons and the glial cells from ALS patients with the fused mutation. Now, another subsection I wanted to highlight, since we have actually a fair number of publications in this section, I think it's a really powerful study, is really looking at neuronal control of feeding and how it relates to metabolism. And so this is a very nice study that came out of Novo Nordisk, where researchers were examining the FGF family of ligands and receptors. The FGF family of ligands has emerged as a promising therapeutic to reduce body weight and inhibit food intake and increase energy expenditure. But whether the specific FGF receptors, as well as the beta-Klotho co-receptor, are involved in these central actions remains unclear. So the researchers at Novo Nordisk used the RNAscope assay to map the central FGFR KLB system to better resolve the anatomy of this system at the cellular level. And they examined this both in mouse as well as a non-human primate and in human samples as well. So you can see here very nice detection of the FGFR1C receptor as well as the Klotho beta co-receptor. And as I mentioned, the RNAscope assay really allowed them to assess expression in a much greater spatially sensitive manner than other traditional-ish assays. They again furthermore combined this with IF to get that cell type specific expression of FGFR. So they were able to show that FGFR is expressed specifically in mouse astrocytes, which are marked by GFAP. There's another study looking at receptors that are also associated with food intake. This is looking at GIP and GLP receptors. So the ligands, or the GIP, GLP incretin hormones, they play significant roles in keeping blood glucose levels normal by stimulating the release of insulin. And agonists against these receptors have been reported to suppress food intake. But their full therapeutic potential has remained obscure because there's been ambiguity over the role of GIP and GIPR in obesity. And so the RNAscope technology was used to characterize a previously unrecognized population of hypothalamic cells in both the mouse and human brain that co-express the GIPR and GLPR receptors. And when these are stimulated with agonists, they can potently reduce the food intake. So you can see here on the bottom of the screen that the multiplex fluorescent assay was used to identify cells that co-express both of these receptors in the ARC and DMH regions of the mouse hypothalamus. They then went on to also look at these receptors in the human hypothalamus as well, excuse me, and also found co-expression of these genes in cells, both in the mid-hypothalamus and the posterior hypothalamus. Now, this is a unique study looking at a particular gene that can regulate locomotor activity. There's a gene called FTO, which is the fat mass obesity-associated protein. That's an obesity risk gene that is also associated with locomotor activity regulation. But how this gene functions in neurons that control voluntary movements, which are specifically the medium spiny neurons, has remained unclear. Now, it's well known that the MSNs have are marked by two different dopamine receptors, the D1 MSNs, which are the direct pathway and can facilitate movement, and then there's the D2 MSNs, which is a more indirect pathway and can suppress movement. And these researchers from Jens Bruning's lab found that FTO regulates physical activity more so via the D2 MSNs in the brain. And so the multiplex assay was used to confirm expression of FTO in the D1 and D2 MSNs. And then they also deleted the gene in these cell populations to confirm that. And they show that indeed there's a correlation with increased locomotor activity in mice that lack the FTO gene, specifically in the D2 MSNs, but not in the D1 MSNs. Okay. Now, this is looking at that control of impulsivity, and they found a new role for a particular gene, MCH, or melanocortin hormone. We know behavioral impulsivity is common in many psychiatric and metabolic disorders, but it's important to understand the substrates that can regulate this impulsivity that can really help improve quality of life for individuals struggling with these disorders. So these researchers found that MCH signaling in the ventral hippocampus can increase impulsive responding and impulsive choice. So this data can really broaden the potential of targeting the MCH system for obesity therapeutics. The RNAscope assay was utilized to identify cell types that express the MCH receptor, MCHR1. They found that the majority of MCHR1 neurons co-localize with VGlut1, so they seem to be more predominantly expressed in glutamatergic neurons, but a fair percentage, 44%, were also found to express the GABAergic marker GAD2. And then by combining the ISH with IF, they revealed the spatial relationship between the ligand and the receptor. So if you look at the bottom of the screen, in red, we can see the receptor identified by RNAscope is in proximity or co-localizes with cells that are expressing MCH1. And then finally, for this section, I wanted to touch upon the vagal sensory neurons that are involved in feeding. This is a publication that also just came out a few weeks ago that created a cell atlas of the vagal neurons that are associated in feeding regulation. So as we know, energy homeostasis really requires the precise measurement of quantity and quality of ingested food. The vagus nerve innervates the gut and can detect those stimulatory cues from the gut. But the key neurons involved had remained unknown. The researchers from UCSF, again, took advantage of single-cell RNA sequencing and cataloged all of the sensory neurons in the vagus nerve to identify the populations that innervate the GI tract and can suppress hunger. And you can see here, it's a very nice combination or a comparison of the single-cell data, which is on the top row, with the RNAscope data, which is in the middle row, and then the bottom row shows a Venn diagram, which shows the overlap of gene expression or cells expressing these particular genes. And so you can see very nice correlation between the single cell data and the RNAscope data. And the two together show very nice vagal subtypes that have unique morphologies and innervation patterns. It really reveals an unexpected role for intestinal mechanoreceptors in the regulation of feeding. So now I'd like to switch gears a bit and just discuss some publications within the immuno-oncology realm. And so the first one I'd like to highlight is a lovely study that came out of Vanderbilt University that looked at the TCR repertoire during disease progression. There was these researchers had a patient who had melanoma. They were being treated with anti-PD-1 therapy. And then during the course of treatment, they developed a neurological adverse event known as encephalitis that was fatal. They did molecular profiling and RNA sequencing analysis throughout the course of development of disease progression. So you can see here as they progress through disease, they looked at the T cell receptor sequence, amino acid sequence, and found that there was one that was really most highly represented in the inflamed brain, or really at the terminal disease progression, and that's the amino acid sequence shown in red. And the clonal population of T cells that seem to be at the site of inflammation. So these researchers took advantage of the BaseScope assay, since it can interrogate short sequences, and identified the TCR sequence at the RNA level in the brain tissue, or inflamed brain tissue from this patient. So you can see the TCR clone is shown here in red. They combine this with IHC for either CD8 or CD4 to identify which specific T cells are expressing this T cell receptor. And they found that the TCR signal was co-expressed in cells that are CD4 positive, but not in the CD8 positive cells. So it's a very interesting study, first one to identify the T cell receptor in the tissue context. This is an interesting one that looked at how microtubules can affect PD-1 expression. So despite the clinical success of many blocking inhibitory receptors such as PD-1, the mechanisms that control the expression have not really been fully elucidated. And so these researchers identified a post-transcriptional regulatory mechanism that can control expression of PD-1 in T cells. And they revealed using RNAscope combined with IF to show that PD-1 mRNA is associated with microtubules, which are marked by tubulin. So you can see this here on the bottom of the screen. You see in untreated samples, PD-1 and mRNA and PD-1 protein are co-localized with the microtubule, which is marked by tubulin in red. But upon treatment with a microtubule inhibitor, you lose the tubulin, you have less PD-1 protein, but then you actually have increased PD-1 mRNA. So it seems that PD-1 mRNA is really associated with the microtubules in activated T lymphocytes. This publication examined the gamma delta T cell subpopulation. They found basically the gamma T cells, we know that they're important in mounting a response against cancer, but one of the early T cells to infiltrate the tumor site and secrete pro-inflammatory cytokines. However, the effect of aging on this gamma delta T cell population is really not well known. So these researchers found that the composition of the gamma delta T cells in the lymph nodes can change in aging mice. And they also found that there's an increase in tumor-promoting IL-17 producing cell gamma delta T cells, and that's what's shown here. You can see the red is marking the constant region of the T cell gamma receptor, and then the yellow is the TRGV6 probe, and that's indicating the IL-7 producing variant as well, shown in white. So you can see how there's an increase in the young mice, there's an increase in the cell population, but the older mice have reduced, reduced gamma delta 17 T cell and IL-7 expression. Now, in this publication, these are researchers that identified LIFR as associated with pancreatic cancer. PDAC, or pancreatic ductal adenocarcinoma, has a dismal prognosis largely due to inefficient diagnosis. And the activation of prostate stellate cells and the development of dense stroma are really prominent features that account for this aggressiveness. There's an interplay between the stromal cells and the cancer cells that can really facilitate a vicious cycle to exacerbate tumorigenesis and drug resistance. These researchers from the Salk Institute found that LIFR is a key paracrine factor that's released from the activated stromal cells and acts on the cancer cells to promote tumor progression in pancreatic ductal cancer. And so we can see here very nicely that RNAscope duplex assay revealed the cells that are producing LIFR and the cells that are expressing its receptor. So they show in the on the left-hand side of the screen, KRT19 is marking the tumor cancer cells. We can see LIFR is really more prominently co-expressed with the, it's not expressed in the tumor, it's limited to the stroma, versus its receptor, which is actually more prominently expressed in the tumor cells and not in the stromal cells. And the study from the authors show how it underscores how a better understanding of cell-to-cell communication within the tumor microenvironment can suggest novel strategies for cancer therapy. This publication from researchers at the Broad Institute examined glioblastoma cells at the single cell level, and they identified that glioblastoma cells actually have different cellular states. They did this identified 4 cellular states. They found that these glioblastoma cells can either belong to a specific cellular state or it can exist as a hybrid of more than one state. These subtypes were identified in patient samples and were consistent with the subtypes that were also found in the TCGA database. And so by using the duplex assay, these researchers could really get visualization of these different cell states and their distribution within human glioblastoma patients. And that's what's shown here on the right-hand side of the screen. And then on the bottom of the screen, you can see quantification of those various hybrid cells that were identified by the RNAscope assay. And finally, for the oncology section, I wanted to highlight, again, another example of splice variants. In addition to the brain having the highest abundance of splice variants, tumors also have a high abundance of splice variants, and they can be associated with disease progression or drug response. The HER2 splice variant, which is known as HER2 delta 16, is characterized by the deletion of exon 16, and it's associated with HER2 positive breast cancer aggressiveness, stemness, and susceptibility to Herceptin. However, it's been very challenging. There's been limited tools that allow one to interrogate these splice variants in the tumor context. And so these authors took advantage of the BaseScope assay, since this assay can now look at those splice variants in the tissue context, and they were able to discriminate the HER2 delta 16 isoform shown here on the far right side of the screen in comparison with the wild type HER2. And they examined this in a wide range of cancer types, including breast cancer, gastric cancer, and colorectal cancer. And they found that there's a positive correlation where an ISH score of 2 or more for HER2 delta 16 correlated to progression-free survival of greater than 12 months after Herceptin treatment. Now, the last section I'd like to touch upon is cell and gene and drug therapy. So in this section here, this is a publication that was came out just a couple months ago, was also presented at SITC recently, looking at the detection of modified TCR T cells in tumors. So the cancer testis antigen NY-ESO1 is highly expressed in synovial sarcomas and is a promising therapeutic target. So researchers at Adaptimmune showed that these TCR T cells that target NY-ESO1 traffic to the tumor microenvironment, and they can maintain cytotoxicity in synovial sarcoma patients. RNAscope assay was used to actually specifically target and detect these TCR T cells in the tissue context. They performed this on 202 patient biopsies following infusion, and they confirmed the persistence of these TCR T cells long after infusion. And that's what's shown here. You can see nice detection of the SPEAR T cells, which are shown in red, or the TCR T cells, and how they're distinguished from the endogenous CD3 positive T cells shown in green. Now, cellular therapies have mainly focused on the T cell response, but there has been some limitations to them, especially because of toxicity due to the cytokine response syndrome. So natural killer cells have come onto the scene as having anti-tumor properties and can potentially be utilized as an alternative to T cell therapies. So researchers at Innate Pharma generated NK cell engagers, which bring tumor cells and NK cells together to trigger tumor cell destruction. And the RNAscope assay was used to detect an NK cell receptor, NCR1, which encodes the gene or the protein NKp46. That's shown here in the upper right portion of the screen, where mice that were treated with the NK cell engager with NKp46 had increased NK cells in the tumors. Now this is switching gears a little bit and now looking at CRISPR gene editing therapy. So LCA10 is a severe retinal dystrophy that's caused by mutations in the SEP290 gene. Researchers at Editas created a compound, EDIT-101, which is a candidate genome editing therapeutic for SEP290 for LCA10 retinal dystrophy. The RNAscope probe was used to target a portion of the AAV virus, shown here on the screen. They targeted the portion that was driving SA-Cas9 expression and did this to show cell type specificity of the AAV virus that was driving expression of the edit. And what this really shows very nicely, they did this in non-human primates. They show that this is the ability to edit the genome in primate somatic cells, unlocking the potential of gene editing for human therapeutics. And then the last example for this section I wanted to touch upon was now also examining the cellular localization of drug targets. So particularly for particular receptors or targets of drugs, we have to have a very thorough interrogation of where these genes are being expressed. Particularly mTORC1 has been linked to several medical conditions that are associated with advanced aging. And cytoplasmic proteins cestrin-1 and cestrin-2 can modulate mTORC activation. The researchers at Navitor had found a compound that can bind cestrin-2 and activate mTORC1. And the duplex assay revealed detailed neuronal localization of both cestrin-1 and cestrin-2. And really, so this overall descriptive study here really supports the goal of developing an active mTORC activator in the CNS that can target specifically the cestrin-1, cestrin-2 pathway. I have two final sections I'd like to touch upon. This one is also very important for application of the RNAscope assay, and that's stem cells and development. So I just want to highlight a couple of key papers in this research area. This is one that was looking at the liver epithelium during homeostasis and regeneration. The liver is one of the few tissues that can substantially regenerate after injury. And these researchers found that there are two major cell types, hepatocytes and biliary epithelial cells, that really play an important role in regeneration. And using single-cell sequencing, they found that the biliary epithelial cells actually have a high amount of heterogeneity that wasn't previously appreciated before. And so they used RNAscope to validate that transcriptional heterogeneity. They found that these biliary epithelial cells were really differentiated by a YAP target gene signature that included expression of CYR61, which is shown here in red on the bottom of the screen. And this appears to co-localize with markers KLF6 or HES1, as well as the keratin marker, which is a marker of these biliary epithelial cells. That's a normal liver. They then went on to go look at this in an injury model of the liver. And they do find that under homeostasis, YAP targets, KLF6 and CYR61 are only expressed in a very few number of these biliary epithelial cells. But upon injury, those two cell populations increase. So KLF6 and CYR61 both increase in expression. And the number of cells that co-express these genes also are increased as well. Now, in another tissue, in the intestine, the stem cells during homeostasis are really marked by LGR5 positive crypt cells. And during homeostasis, these are the ones that really seem to drive turnover of that intestinal epithelial population. But upon injury, those LGR5 positive stem cells are actually lost. And however, the epithelium is still able to recover. There has to be another population of stem cells that can drive this renewal in response to injury. And so researchers used, again, single-cell RNA sequencing to profile that regenerating intestine, and they identified a novel cell type, which they termed revival stem cells that are marked by a gene called CLU. And that's shown here on the bottom of the screen. You can see the distinct clusters of cells that are enriched following irradiation, cluster 18 marked in red, and this is marked predominantly by CLU, but also as other genes as well. And RNAscope was used to confirm in the normal intestine that this population does exist, but to a very small extent. The LGR5 population is still much more abundant. However, on the right side of the screen, in response to injury, you now start to see expansion of the CLU-positive cells shown in green. So it indicates that the CLU-positive cells are really the ones that contribute to intestinal cell renewal after damage. Now, also continuing on, looking at the colon, this is a unique, another unique tissue. It has epithelial cells, there's renewal as well, but it seems that there's also evidence that stromal cells can contribute to the intestinal cell niche, but those markers and the signals have not been well defined. So the researchers here identify that CD90 positive fibroblasts are located in close proximity to the stem cells in that niche, and they can support growth and proliferation of these stem cells. And so RNAscope was used to, in combination with immunofluorescence, to reveal the spatial localization of these CD90 positive fibroblasts. You can see they're shown in red. They are located at the bottom of the crypt. They co-express with BRDU, so they are expressed in cells that are marked by BRDU at the base of the crypt, so they're expressed in proliferating fibroblasts. And then they also show that these cells are also producing SEMA3 or semaphorin 3, which are soluble mediators, and they're involved in the signaling from the fibroblasts to the stem cells. Now, an interesting model of early regeneration is the salamander. And so researchers from Doug Melton's lab utilized this animal model as a way of looking at regeneration. They really want to understand the mechanisms behind the formation of a blastema, which is basically the cellular structure that is basically it starts to form distal to the amputation site. And this could be by looking at the mechanisms that are behind blastema formation, this could provide insight into human regenerative potential. So these researchers found that blastemal progenitor cells are early mediators of immune support via the IL-8-CXCR1-2 signaling pathway. So RNAscope was really used to identify the transcriptional landscape. You can see here through sequencing analysis, all of the genes in the heat map. RNAscope was used to validate this gene expression during various days post-amputation in the salamander. And then they go on further to even do more of a descriptive study here, looking at the IL-8 expression as well as the IL-8 receptor, CXCR1-2, and identifying the cell types that are expressing both of these genes. And the last section I wanted to highlight here is this really nice study looking at ciliogenesis. Ciliopathies are due to diverse gene mutations that can result in primary cilium malformation or dysfunction. These authors identified the secreted metalloproteases, ADAM-TS9 and TS20, as being required. They can act intracellularly, and they're ciliogenesis regulators. And RNAscope was able to look at that spatiotemporal expression pattern of both of these genes and the effects of their deletion during development. Now, within a subsection of developmental biology, I think the whole mount technology is a really unique application that's really well positioned for developmental biology, and it's something that has been applied using the RNAscope assay. And I want to show some really nice images from publications that utilize this. So this is looking at the Drosophila retina. These researchers identified cell type expression in whole Drosophila retina in order to study the light-dark clock cycles. And really just want you to appreciate how they can get that spatial resolution of cells that are expressing either HIF-CL1 or ORT. Genes that are involved in the light-dark cycle, and they show it's specifically expressed in either photoreceptors or if it's expressed in interneurons. This is a publication here where they actually combined the RNAscope-ish with immunofluorescence on whole mouse skeletal muscle fibers. And so they're able to take the whole individual muscle fiber, examine it for both RNA expression and protein expression. You can see here, particularly on the right-hand side, the really nice striations marked by CAV1, by IHC, and then looking at the gene expression for either PAX7 to look at the resonant stem cells, and MyoD to look at the more mature myogenic cells. And then finally, this is a publication that came out of the Gladstone Institute at UCSF that was looking at heterogeneity during early embryonic heart development. Dysregulation of cell type specific gene networks can result in birth defects, which can affect 5% of live births. The congenital heart defects are the most common malformations, but really the transcriptional changes that lead to these defects have not been well characterized. So by using single-cell RNA sequencing, they were able to find and identify the early cardiac progenitor cells that are there during normal and abnormal cardiogenesis. So some really beautiful images of the whole mount mouse embryo, really focused on the cardiogenic regions, which you can see here on the right-hand screen, when they're able to do multiplex RNAscope staining to look at that gene expression. And these are additional images to show that nice spatial detection of the gene during cardiac development, and then also on the right-hand side, looking at the section tissues. And then finally, the very last section is looking at infectious diseases, which is, again, really another important application here for RNAscope, since we can interrogate both positive and negative strands of these viruses. And so this is an important virus that's been emerging recently. It's a Nipah virus, which is a negative sense RNA virus. It's an emerging, highly lethal zoonotic virus that can cause severe brain encephalitis. There was an outbreak about 20 years ago in Malaysia where the fatality rate was 40%, and then more recently in May, there was an outbreak in India where the fatality rate was 91%, so it's highly fatal. Those humans that do survive have relapsing encephalitis. So these researchers looked at a non-human primate model to see if the Nipah virus persists. And shown here on the right-hand side, you can see indeed the ISH is able to detect the Nipah virus in the brain, and they looked at both the positive or antisense and the negative sense strands. And by doing both of this, it really indicates active Nipah virus replication and shows the persistence of these brains in non-human primate survivors with a previous infection. In addition, as we well are aware of the Zika virus epidemic, it has been known that it can be sexually transmitted. However, the tropism in the reproductive tract, particularly the female reproductive tract, had not been known. And so researchers at Washington University identified a role for interferons in controlling Zika virus infection. And they show that interferons can actually inhibit Zika virus infection in the female reproductive tract. And that's what's shown here on the right-hand side. We can see from mice that were infected with Zika virus, there is strong detection in the vagina. However, after treatment with interferon gamma, this signal is significantly reduced, if any is even present. And then finally, this is another interesting virus, Rift Valley fever virus, which can cause severe disease in livestock, but it can also be transmitted to humans. And a subset of people that are infected with this virus can develop encephalitis, but the pathology in the brain is really limited. So these researchers looked at a rat brain model of this viral infection to identify what cell populations are responding. They found that it's IBA1 positive macroglia that can increase in response to this infection. And they examined this by combining not only RNAscope for mRNA with immunofluorescence for the protein, and they also used neurotrace to mark the neurons. And with that, we've covered quite a few papers, but that was really just kind of the tip of the iceberg for all of the stellar RNAscope publications that we have this year. Again, we had almost 800 new papers in really many exciting new research areas and really innovative applications of RNAscope. And I think these publications really highlight the sensitivity and specificity of the assay, showing the multiplexing capabilities, and now with high-plex, we can now detect up to 12 targets. With the BaseScope assay, we can interrogate those RNA species that haven't been able to be interrogated in the tissue before, such as splice variants and short sequences, and it can be combined with ISH or with IHC or immunofluorescence in order to get simultaneous RNA and protein detection. And so with that, I'd like to really thank all of the researchers who have published with RNAscope. Again, we thank you for your hard work, your stellar work. It's very impressive. So we are out with the 2019 publications, but we are very excited to see what 2020 has in store and what you as researchers are going to do with the assay. And so with that, we'd like to wish you a happy holiday. Please feel free to send any questions to us so we can answer online and by email as well. And please visit us on the web or direct any questions to the emails shown here on the screen. Thank you very much.